Good morning. Welcome to the first webinar in a series for the project Developing Sustainable Solutions to Cassava Disease in Mainland Southeast Asia. The topic of today's presentations are Keep the Faith, Progress in Developing Commercially Viable CMD Resistant Varieties for Asia. I came up with that title sitting and feeling a little bit depressed about the current situation, but then hearing the reports and seeing some of the pictures of what's happening in the field, I was reinvigorated and encouraging everyone to keep the faith that a solution will come. There's a number of events in your life where you remember exactly where you are when it happened, and for me, on December 11th, 2015, I was in Lima, Peru, and the director of the Cassava program at CIAT at the time came to me to tell me that CMD had been found in Cambodia. I just finished a presentation describing the economic value of cassava in Southeast Asia and how the different countries were all linked through the value chain. So I knew how serious this would be, the impact at different levels along the value chain. So from those initial reports, in Cambodia was first found in 2015. You can see now we have up to 59 provinces with some level of CMD infection. It's a little bit hard to estimate the total number of hectares that are infected, but I think it's safe to say that there's between 150 to 200,000 hectares currently with some level of CMD infection. Again, it's difficult to kind of get an idea of the yield impact and the production impact, but a good place to start is to look at Cambodia, where there's limited domestic processing utilization. So you get an idea by looking at the export and trade data of what's happening. And you can see in this slide on the left, from 2017 to 2020, up until June, there's already a reduction in the value of imports into Vietnam from Cambodia of around $126 million. On the other side, on the western border, the reduction has been much less significant, and there's still areas and pockets in the west with limited or no CMD infection, but you can kind of get an idea of where things are going for other countries as the infection takes off. Of course, this flows through to the whole industry. On the left here, you can see the price of roots in Vietnam compared to Thailand. So the blue line on top, you can see the price of roots in Tay Ninh here, and the red line here is the price of roots in Thailand converted to 30% starch content, which is the benchmark in Vietnam. So you can see in Tay Ninh, they're paying a considerable premium over what the Thai price is. So up to $40 a ton extra for the roots, just because now with that level of infection in both Cambodia and Tay Ninh's local area, and the high level of capacity for processing, there is fierce competition to get access to those roots. On the other hand, you have on the right-hand side the FOB price of starch, again comparing Bangkok FOB to Ho Chi Minh FOB. So you can see here the challenge for those processors in Vietnam. They're paying more for the feedstock and then receiving less for the final product. There are a number of factors that need to be also considered in terms of deductions due to quality, and also the added income from using the residue for livestock feed. But I think you can all see that the industry is also suffering greatly as yields decline due to disease. Of course, for us at CIAT, and I think a number of people, the main thing that we're most worried about is those impacts at the farm level, with farmers and communities really suffering as yields are declining. We recently had a survey in the east of Cambodia, and when I got the data from the team, the yields were actually so low that I had to ring them up and just confirm. Again, this is not all due to CMD. There's been impact of drought and poor germination, but it's certainly contributing to lower yields. And I think it's even more important to think about this in the current situation with the COVID migration and travel restrictions. A lot of these farmers' secondary livelihood option was either to travel to cities or into Thailand to seek out off-farm employment. And currently that's not able. So we'll see fairly significant impacts on households this year. So looking at those statistics and numbers, it's quite easy to get quite depressed about the situation at the moment. Large impacts at the farm level flowing through to industry and then national incomes. But I hope today, when you see the presentations, you see that we are making progress in terms of the development of resistant varieties. And while that solution may not be here tomorrow, it's certainly in the future. So I would ask everyone in the industry to keep the faith and know that the teams in the various countries are working very hard to develop these solutions. So I'd firstly like to thank the support for this project, the Australian government through the Australian Centre for International Agricultural Research, and also the CGR research program on roots, tubers and bananas that are the primary supporters to the project establishing sustainable solutions to cassava disease in mainland Southeast Asia. Today you'll hear reports from Thailand and Vietnam, but I'd also like to thank all the partners involved in the project and the considerable in-kind support and dedication that we receive from them in terms of the activities that are underway. So you'll hear today about the breeding activities, just to outline what that objective is, to enhance capacity and collaboration between breeding programs in mainland Southeast Asia to develop new product profiles for commercially viable cassava varieties by identifying and incorporating known and novel sources of resistance to cassava mosaic disease, but also cassava witches broom disease that is also a significant problem in the region. It's great to have this seminar because it really highlights that collaboration and sharing information, and also to have some industry members join us today because we really do need to make sure that we're developing products that are commercially viable so that the cassava sector and cassava farmers can continue to compete on a global market. So again, this is the structure of the research project. Today we're really looking at the pink area, and we'll be hearing around the different sources of genetic material that has entered into the country. So we'll be hearing about all this material here that is feeding into the screening and breeding. We hope to have ongoing seminars looking at how that will flow through to the seed system in the coming weeks, and also an update on what's happening in terms of surveillance and diagnostics. But with that, again, I'd like to thank everyone for joining today. You will be able to download the presentations at the project website. And also, please do join us on Facebook to share information and ideas and keep up to date with what's happening with each of the teams. So this morning we have three very interesting presentations. We'll start off with Dr. Xiaofei Zhang from SIA, cassava breeder based in Colombia, who will be outlining the different sources of resistance that have been introduced to Southeast Asia. We will then hear from Professor Ed Sorobol, who will present on behalf of the team from Kasetsart University and TTDI on the activities in Thailand. And finally, we'll have Dr. Vu Nguyen, who will give us an update on the screening activities that are happening in Vietnam. We do have plenty of time for discussion after the presentations, so please stick around, listen and contribute. And with that, I'll pass it over to Xiaofei, who will lead us with the first presentation. Thank you, Xiaofei.
I believe you can hear me well, right? And also see my screen well?
Yes, very clear. Thank you.
All right, thank you, Jono. Let me put my slide. All right, thank you, Jonah, for organizing this webinar. It's really excited to show the activities in Southeast Asia. So I'm going to mainly talk about the source populations for CMD resistance we introduced to Southeast Asia. So I think first, let me briefly introduce about the Cassava program. So the program leader is Luis Gusto, and we have 6 components. So one is about breeding and genetics analysis, and we have scientists working on agronomy and physiology, also plant pathology, and scientists on seed system. And we also have food scientists again, and again, Jono working on value chain and almost everything there. So for the breeding program, we have Nielsen working on cross industry. He is in charge of meeting crosses at CEAT in Palmyra. So Sandra is in charge of the field trial in Palmyra, and Jorge Wan is in charge of the yield trial and the coast. So where the major cassava production area in Colombia. So the environment might be quite similar with Asia. And Dewey is the scientist overseeing the yield trial in Vietnam. And the list base is the data manager to manage all the data from the breeding program. And in terms of the cassava breeding in CEAT, so we work on, we kind of serve the cassava community in Latin America, in Africa, and also in Asia for sure. So for Latin America, we provide trait donors and also improve the population and release variety for three different products. Here, high beta carotene and also normal or conventional cassava with good clean quality and high stable dry matter. And for South Africa, we mainly work with we provide trait donor and improve the breeding population for the same 3 products. And for Asia, we mainly work with different national programs to provide trait donor and improve the population for high and stable dry matter cassava. For sure, for now, it has to be CMD resistance. So about the source population for CMD resistance, I summarize them here. There are about 6 populations. So the first 2 were introduced into Asia years ago. So 5 ITA clones, they show good CMD resistance. They were confirmed in the field in Asia. And also some C clones, C33, C39. C33 is very good. C39 is partial resistance, and CME3 seems not very good based on the data. And the other 4 are kind of new populations. So in Vietnam, from the variety collected from the farmer's field, we find 10 varieties show good resistance, and also introduced the 102 CEAT clones. These clones show CMD, might have CMD2 genes based on the SSR marker. And another population is the seeds introduced from Hawaii. So some progenitor from ITA, they might provide CMD resistance. And the last population is a breeding population. We are going to test genomic selection and also identify major QTLs for CMD resistance, design diagnostic markers, and also identify QTL for dry matter and also plant type. So for the 5 ITA clones, you can find more information or detailed information in Cassava base. So this Cassava base is supported by the Next Gene project. So the project is funded by Bill Melinda Gates Foundation. So the Cassava base is designed for breeding trials and also agronomic trials. So we can see if we can design the trial, we can upload the data, and also analyze the data in Cassava base. So for example, here this is one of the 5 IT clones. If you search the clone there, you can find the phenotype data, the pedigree. And here I show the CMD score. So the average is 1. So that means it show very good CMD resistance in Africa. And if you want to learn more about the Cassava base, so here is some tutorial. So at CEAT, we also start using Cassava base. And for the resistant variety from Vietnam, so 5 years ago, CEAT and Hang Lok Agricultural Research Center and also Root Crop Research Development Center, they did a survey in Vietnam. So they harvest samples from farmers' field and also from different center. So in total, more than 3,000 samples, and we find 109 unique varieties. We put in 2018, and we collected the CMD data, germination, and also yield data. So we find some clones show very poor germination. So after removing those plots, so we still have 94 unique varieties. So we analyze the data. I summarize the result here. So there are CMD score, 6 weeks, 3 months, 6 months, 10 months germination, and also yield per plot. So you can see the CMD score increase from 6 weeks to 10 months, and also the heritability increase. So especially for 6 months and 10 months data, we got high heritability. That means we managed the trial well, and we have high quality data. So we compare some clones show good paired with the resist variety, for example, 50, the number is like this, and also KM419 is like this. But all these 10 clones from early to late show better CMD resistance. And here I want to point 2 clones. For 2, it show good germination and also high yield, and 773 also showed good germination and also the yield is good. But some clones show poor germination. Again, so this is, I believe the CMD score is reliable, but the germination, the yield, since this is only from one location, one environment, so we still need more tests. And another clone I want to point out is this PROR 262 and CR63, this and Thai9. So based on the DNA fingerprint, we find the sample in Vietnam is the same with the clones in the core collection in the gene bank at CEAT. So this Thai9 is from Asia. So the variety name in Asia is Hanatin. I believe a lot of people know this clone. Seems like this clone show good resistance. So based on these results, we kind of believe in the gene bank and see that there might be good some source of CMD resistance. We are planning to screen the core collection. Hopefully we can find some new CMD resistance there. So another CMD resistant population is from CEAT, introduced to Vietnam several years ago. And the last year, AGI, Agricultural Genetics Institute, Dr. Wu, they put the trial, these 102 clones, and several other CEAT clone, 5 IIT clones in a field in Taining. So you can see here, these are some resistant clones, and again, these are some released varieties. The date, I have the data of 1 month after planting, 2 months after planting. The CMD score is green. That means it's almost no symptom there. And here the count means how many plants are there. So you can see the number of plants is pretty high. That means the result is pretty reliable. And here there are 4 IIT clones, also show very good resistance. And then this year in May, Hanlock Research Center and AGI, they established trials in Taining and Dong Nai. So we have 48 clones with RCBD. And next spring we are going to harvest them. We will have a better idea about the yield and dry matter of these resist clones. Hopefully we can find some good ones. And from the resistant clones, we screen them at CEAT Palmyra in the tissue culture lab. So there are 28 clones. They are negative in different virus and disease. So they are ready to share with TTDI, also other breeding programs if we got the request. So the next source publication is the seed from Hawaii. So in the Next Gen project, CEAT and ITA, we provide germplasm or progenitor to Hawaii. There they make crosses, and then they send the seeds back to ITA or to CEAT. So they have some leftover seeds. We request them to send to Asia breeding program. So Hanlock Research Center, they got the import permit early. So I believe they already received the seeds, about 5,000 seeds. So there should be some good CMD resistant clone from this publication. And the seeds for Thailand and Lao is still, the breeder in ITA is still working on the list. So, and then the last publication is breeding publication. So at CEAT, we make crosses between high dry matter clones and CMD resistant clones. So you can see C33 here and C413, C90, C39. They are all CMD resistant clones. So from this publication, we sampled about 400 clones. So this is the number of progeny from each family. So about 400 clones, 400 seeds. We plant them in Palmyra at CEAT, and also we send a publication from the same family to AGI. So we are going to genotype and phenotype both publication in two environment, and to identify the QTLs for CMD resistance. Hopefully we can design reliable diagnostic markers for the breeding population. And also we are going to test the genomic selection in CMD breeding. So genomic selection has been widely used in plant breeding like corns and wheat and soybeans. And also in Next Gen, it's been tested in Africa and also in Brazil. So this on the left side is conventional breeding. On the right side is the new germ selection strategy we developed. So basically, we sample about 400 clones or 400 genotypes from the breeding population. It's a very small population compared with the breeding population. So the selection is based on the pedigree. So in the first year, we do quick clonal propagation to get 5 plants from 1 seed and then plant them in the field. So in the next year, we will have enough cuttings to develop yield trial in multiple locations, multiple reps. And we are going to genotype this population. So based on the genotype and phenotype, we are going to develop a statistical model. We call it the germ section model. And for the breeding population, we still plan the big breeding population in the field, and based on the 1 year field evaluation to trim down the population size from 25,000 to 1 or 2,000, then we can genotype the selections and bring the genotype data to the statistical model. And based on that model, we are going to calculate the breeding value to select the best clone to cycle back to the crossing nursery, making crosses, and to develop the breeding population for next cycle. And also we can predict the phenotype data to select the best clones, put them in preliminary yield trial for variety development. So compare with conventional breeding, so you can see the cycle time is reduced from 5 to 3 years. And also we skip the clonal evaluation stage, so it can save 1 year for variety development. And again, we have the genotype phenotype data. We will be able to identify or validate the markers for marker assisted selection, for example, for dry matter, for CMD, for plant type. So again, so based on this strategy, so we have one population in Colombia, and we have another population in Vietnam. So we are going to build the statistical model for Colombia, for Vietnam, and then predict the breeding value, and then make selection based on the model from Vietnam, select the best parent, and then use the parents to make crosses, produce seeds. The seeds will be sent back to Asia. So we are expecting those seeds will have better adaptation to Asia environment. And also we are talking with IITA breeding program to do the similar thing with IITA. So I'm going to stop here. So again, I would like to highlight the 6 CMD resistant populations introduced to Asia. And I also appreciate the great collaboration with AGI, with Hanlock Research, Agricultural Research Center, and also with TTDI for sure, and thanks IITA's great support. And we got great support from RTB and NextGene, Cassava, and also thank the support from ACRAR for support all the breeding activities in Southeast Asia. So hopefully I have some time for questions. Thank you.
Thanks, Xiaofei. We've got time for a couple of quick questions, and then we have longer at the end of all the presentations. So if someone has a burning question they'd like to address to Xiaofei now, please open your mic and ask him.
Hope I'm clear.
Okay, so you can think about questions as we have the next 2 presentations and ask them there. The next presentation is from Dr. Ed Sarabol from Kasetsart University TTDI team. I'm going to share the screen, and Dr. Ed, if you can just tell me when you would like me to move to the next next slide. So if you can try to open your mic.
Yes. Do you hear me?
Yes, we hear you well.
Okay.
Okay.
Sorry, Dr. Ed. Now I'll bring this up.
All right.
Full screen.
Yes.
Okay, over to you, Dr. Ed.
All right. Thank you so much. Good morning, everyone. I hope all of you and your families are safe from COVID. The names that appears on the slide is our team for this project. The information that I would like to share with you this time is only from breeding aspect. Okay. Next slide, please. We did the field trial in 3 locations. The course of the experiment or the trials cover April last year, and we harvest this year's around April or May. The 3 locations were Suwan Farm and Hoi Bong TDI field. These 2 locations are located in Nakhon Ratchasima province, which is the home of the cassava in Thailand. And another one is Khao Hin Son field trial, which is located in Cha Cheung Sao province, close to Sa Kaeo, near the border. But these 3 locations, we did not have disease pressure. We want to test for the performance of our commercial varieties and also the resistant clones that we have on our hand before we start the projects. The next slide, please. Okay, from Suwan Farm. This slide may be a little bit small for you, so I asked Dr. Chalermpon to chop it. Okay, so next slide we can see. Next slide, please. Next slide, please. Okay, better. This is from Suwan Farm. Okay, starch content. The starch contents this year from these 3 locations were not so good because of the drought that we have, and before we harvested the cassava, we received some amount of rainfall. Okay, so you'll see that the TME3 number 9 and number 10 C33. Okay, these are 2 clones that resistant to CMD. The starch percentage is about 10%. And the 3 columns, say R1, KU50, and Hoi Bong 80, we compare the clones that we tested with Rayong 1, KU50, and Hoi Bong 80, so you can see the numbers. Next slide, please. So we are interested in the yield. Suwan Farm, the soil is very fertile, so yields for these cassavas are very high. Again, looking at number 9 and number 10, they are both resistant to CMD, but the yield performance is not so good when we compare with our commercial varieties. TME3 is about 6 tons per Thai unit per rai, and C33 is only 2.3. Next, please. And at TDI, again, because the slides, the numbers you see here may be too small for you. So we go to next slide. So yes, you see that the yield performance at Hoi Bong is lower than Suwan Farm. Again, because we don't have, TME3 and C33 is at the lower positions in terms of yield performance. Only 2.9 for TME3 and 1.1 for C33. And also for starch content, for next slide, please. Starch content somehow is better at TTDI, but for TME3, the starch content is around 20. Next slide for Khao Hin Son. Again, next slide, please. Okay, you will see that the yield performance of TME3 is the lowest. So first, we think that if the yield performance for the CMD resistant clones, if they are comparable to commercial varieties, maybe we can go ahead and use these clones or varieties as a stopgap for farmers in Thailand. But as you can see that the yield performance is not so good, so maybe we have to go for KU50 or Rayong 72. Okay, again, looking at the starch content, it's not so good. These three locations, we did not have disease pressures. Okay. But for the next trial, next slide, please. The next trial, we put the trial into Sa Kaeo province where the disease is very severe. Again, the start and the start of the trial is in May, and we harvested the cassava around April. The yield performance is not so good. However, KU50 showed the best in this location. And again, TME3 is the lowest. Starch content is very low in this location. And looking at the number 89, which is very susceptible to the CMD. Yield is still okay at this location this year. So from these results, we put the trial again at near this location, but we have to move to another field because we would like to expand our trials in Sa Kaeo. So we moved to another location, which is about maybe 5 kilometers from the old locations. And again, we modify our trials to get better results from this trial. And maybe next year we can share with you what we get from the trial. Next slide, please. The next three slides show you the disease incident, disease severity, and disease index. These three slides, I would like to ask Dr. Wanvisa to explain the last three slides. Dr. Wanvisa, please, can you join?
Yes, thank you very much, Dr. Ed. So for this slide, our team observed the CMD incident every month, start from 1 month until 9 months after planting. So from this slide, we found the disease incident in KU50, the lowest when compared with another. The second one that kind of also the disease incident low, that is Huaibong 60. Next slide, please. So for the disease index, we found that this disease index, actually we look at the 9 months before harvesting. So the results show that KU50, Layong 11, they has a low disease index is about cannot find about for KU50 about 25% disease index, and Layong 1 is about 30%. So you see the relationship between KU50 and Layong 1. That what I know from the breeder. They told me that Layong 1 is a mother of KU50. So for the TME3, the disease incidence is about 30%. So, but then we noticed something that TME3 in the beginning of the season, we see the symptom, severe symptom, and then it seems to be like TME3 can recover when they are older. And as you know that Dr. Ed already presented in the beginning that the product of TME3 is not so good compared with KU50 and another variety, that commercial variety in Thailand. Next slide, please. So for observe the severity of the variety we plant, we use the score from 1 to 5. That's based on IITA recommendation. So in this slide, we show that the severity of CMD in Thailand average about 2.5 until 3. This is like a moderate severe for CMD in Thailand. Next slide, please.
We have only 3 slides, Dr. Wanvisa.
Yes, for my part is already done. Thank you very much.
We have introduced the CMD resistant clones from IITA, and we got 5 clones, and now they are in the fields for increasing the number of stems. And also we are about to introduce in the near futures, clones from SEAT and also from India. Now we are waiting for sending out the import permit for the 2 organizations. Thank you very much, Jonah.
Thank you, Dr. Ed, and thank you, Dr. Wanvisa. Very interesting results. Some show consistency with what we've observed in Cambodia with the high susceptibility of Rayong 11 and variety 89. I think it may come out in Vu's presentation. One of the confusions for me still is the difference in performance of KU50 that we're seeing in Cambodia and Thailand compared to the situation in Tay Ninh at the moment. So maybe Vu can comment on that during his presentation. So at this stage, is there any questions to Dr. Ed and King? Otherwise, we will hold them over. Okay, in that case, Vu, please take over. So introducing Dr. Vu Nguyen from the AGI. He's the group leader of the national lab there. He'll be presenting on behalf of the team at AGI. Thanks, Vu.
Yep, thank you, Jono. Good morning and good evening, everyone. Can you hear me clearly? And can you
Very
see
clearly,
the slide?
thank you.
Yes,
All good.
yes.
Okay, thank you. So on behalf of the team here in Vietnam, AGI, I'd like to present some of the updates on our CMD resistant screening in the project, establishing sustainable solution to cassava disease in mainland Southeast Asia. So the activities that we've been doing so far, including import the cassava accession and varieties from CIAT and from IITA, and then we did the propagation for providing this material to our partners, both in Vietnam as well as to other countries. And we screened our materials that we've imported, as well as the material from Vietnam for CMD resistance and for yield performance, and also breeding for new CMD resistant varieties. So far we've imported 102 clones from CIAT with CMD resistant parents, 5 varieties from IITA, and we also received more than 400 seeds from CIAT that Xiaofei already mentioned in his presentation. So from this material, we did in vitro propagations of the 102 clones from CIAT. From this, we multiplied more than 200 plants per clones for field testing. So this way we can test them quickly without having to spend 1 year in the field to increase the plant materials. So this was quite quick. We received this material from 2019, and we already have them in the field one season. And for the 5 varieties from IITA, we received them a little bit earlier in December 2018. And from this material, we propagate more than 1,000 plants per clones for field tests and for distribution to our partners. And from the seed from CIAT, we germinated them and we propagate for field tests. So as you can see here, the plants that we did in vitro propagation, and then we transfer them to soil pots. After 2 months in the soil pots, we can plant them in the field. And you can see here the plants ready for transportation from AGI to the field test area in Tay Ninh in the south of Vietnam. So for the clones, we did the first field test in October 2019, and the second one we started in May this year, of 102 clones from CIAT and 5 varieties from IITA. This is the location where we did the screenings in Tay Ninh, which is about 140 kilometers from Ho Chi Minh City in the south of Vietnam. And the period, as I mentioned, from November last year, and at the end of this month or next month, we're gonna harvest them. So we haven't got the full yield data yet. So hopefully by the end of next month, we can have the yield data. As you can see here, I just want to show how the disease spread in the field. This field actually is not from the field that we did for CMD screening with the IITA and the CIAT varieties, but from the local Vietnamese varieties. So most of them did not carry any CMD resistance. And you can see on the screen here, in 1 month, after 1 month after planting, the disease is almost free from disease. And 3 months, you start to have the CMD score of the plants of 2 to 3, which is mild to medium. And then 6 months, you start to see severity in almost half of the field. And by 9 months, you can see the field is completely infected. This is by natural infection with the whiteflies. Because the CMD disease pressure in tanning, it's very high. So this way we can test our CMD resistant varieties. And from the field test, so far we could score 48 CMD resistant clones out of the 102 clones that we received from CIATS. And all of the 5 varieties from ITA show very strong CMD resistance. And they also show reasonable yield, about 27 to 30 tons per hectare. But starch content, it's relatively low compared to our commercial varieties. It is only about 24 to 27%. This is what we score last month, so hopefully it can increase a little bit by the end of the harvesting season. Here I just show you some of the photos that we took. This is TME B419 after 5 months. This is comparing the KM419, which is the best commercial varieties in Vietnam so far, but it is susceptible to CMD on the left. And on the right is we call Hanoi 2 or the IITA TMS IBA 980581 CMD resistant. We show completely no symptoms, CMD symptoms at all. This is the 105, 103, 5 months old in the field. Because we planted 1,000 of these plants, so we can take some of them out and just to have a look at the performance at these different stages. This is another one from ITA at 5 months old. However, we also did confirmation of CMD resistance by grafting, because in the field you can have escape infection by whiteflies, or some of the plants may not be very attractive to the whiteflies. So what we did was we graft the clones of interest onto infected rootstocks, and this way the virus can be transmitted easily to the scions, so we can check for sure if the plant is susceptible or resistant to MD. So as you can see here, this is the scions grafted on the rootstock. Here is the grafting nodes. And here we compare, this is the C33 resistant, which show no symptoms at all. And this one is one of the susceptible, which has the curly leaves and mosaic symptoms. So we could confirm all the over 40 CMD resistant clones. However, this way it's a little bit difficult to do, because you know the grafting of the 2 stems, it's very difficult. We experienced that this one I learned this grafting technique from CIATS back in 2015 when we did the frog skin disease screening. And this way it requires a lot of experience to do it successfully. So we developed another grafting methods. So we don't do in the lab or in the greenhouse, but we do on the field. So this method, it's very simple, efficient, and we can produce the result within 6 to 8 months after receiving the in vitro plants, and we can test up to 1,000 lines at a time without any requirement for skill staff to do this. So these are some of the photos of the CMD resistant varieties from ITA on the large scales. And yeah, these are the seeds that we received from SEATs. So we germinated the seeds in the substrate like this, and then we transfer to the soil pots. And from the material, we receive a total of 42 varieties and a total of 475 seeds. But the germination rate is actually varies a lot between different lines. So as you can see here, up to 100% in some and only like 30 or even 16% in others. So at the end of the day, we could get around 300 plants. So we will continue with this screening first, we will continue with propagation of these plants to increase the plant numbers, and then we plant them in the field as Selfie has explained in his presentation for screening for CMD resistance, and then we can do genomic selections based on this data. So for the breeding, we haven't done a lot in the breeding because we just started early this year. But from last year, we screened our materials that we received from SEAT, and including 2 varieties, 2 clones, C32, no, C33 and C39. So we saw as Thailand team has shown that C33 actually has very low yield, although it's a very good CMD resistant candidate, but has very low yield. But the other one, C39, actually this one is also has CMD resistant, but it's not a total resistant, it's only a CMD tolerance with a CMD score of around 2. So in the beginning, this clone show quite severe symptoms, but as the plant grow, the symptoms decrease, and at the end, it looks quite normal. So a CMD score of only 2. And it has very good yield. We tested, and some plants have the tuber yield of up to 12 kilograms per plant. So we estimate the yield of 70 to 100 tons per hectare. However, it is very branchy and it has low starch content. So because it flowers very profusely, so it's a good candidate parents. And we already produce quite a number of seeds from these clones, this C39, and we'll test them this year. So yeah, this is the end of my presentations. These are the members of our international laboratory for cassava molecular breeding in AGI. And I'd like to thank all our partners from SEAT, from ITA, from FAO, and from Japan, we have the SATREPS and JICA project. From Vietnam, we have our partners in PPRI, in Hưng Lộc, and the plant protection sub-department of plant protection and crop production in Tây Ninh, which help us a lot with the screening in Tây Ninh area. And thank you for your kind attentions.
Thanks very much, Phu. I've got just a quick question. I think I know the answer to. The current trials were established using seedlings, weren't they? Whilst the one that was established in May is now been established with stems. Is that correct?
Yes, the one that we started in May, we use stems. So we collected from the field from last year. So the stem was about like 8 months old.
Okay, so the floor is open to questions to Vu, but also feel free to add questions or comments more generally to the other speakers.
Hello. You hear me?
Yes.
I can hear you, but I'm not sure where you're coming from. Who?
Yeah, my name is with the name Monsak from
Okay. Chiromsa, how are you? Good to see you.
Meeting room in at ETBI in Hoi Mong. Very interesting to see the result from Vietnam. But one thing, I want to share the experience with the condition in Thailand from our try in Sakeo observation in the farmer field in the early heavy inspection in Nakhon Ratchasima. The situation of the CMD in Thailand are very serious in the Nakhon Ratchasima ori, mainly in Nakhon Ratchasima, maybe 80 or 90% of the infected field from one province. The field observation, because in Nakhon Ratchasima they plant a very susceptible variety called the clone number 89. But from our field evaluation, we don't see the much difference in the severity of the CMD among the KU50 or susceptible clone around 2 or 3. The rating of the KU50 with the Vietnamese scientists very similar, around 2.3 or 2 to 3. But the difference is that, you show only the disease severity. But the percent infection, percent infection of CMD, if we check it every month, we can see the difference. The difference between the susceptible clone number 89 and Rayong 11 compared with Kasetsart 50 and Hoi Bong 60. The rate of disease increase in infection rate, very slow. And the final is around 20% infection, but the severity is similar. We don't have the new clone from IITA in the field trials, but we have a clone C33 by grafting. By grafting C33, the disease severity is the same, the same of a report from Vietnam. If we use only the severity, but I still, because we don't have the more information on the experience in the field. In Nakhon Ratchasima area, in some village, if they grow the Kasetsart 50 or Rayong 72 in that village, the disease very mild, not severe. They can survive. If we score as at the end, if we use the severity, all of our KU50 and Rayong 72, all very susceptible because sometimes go to rating number 3, but under the field condition, but some, some in some area with the heavy infection, very severe of the disease. It's maybe Kasetsart 50, maybe get very high percentage of infection. But at the moment, we don't have the resistance. I mean, if we score the rating to be number 1, we don't have yet. We have resistance, but under that variety cannot be accepted under commercial in Thailand. So we try to formulate the recommendation to the industry, and and complement that, how we can stay with the cassava production with CMD with less, damage. So we recommend that we need large amount of the not susceptible clone, not grow first, not grow the susceptible clone. I to find the disease-free that less susceptible, look like Kasetsart 50, Hoi 60, and Rayong 72 into the heavy infection area. Only that, but for the real resistance, But if in Vietnam, if you can show, rather, if we rate the disease, the same system, but if we have the infection rate, maybe we can see the situation when we go out into the field. If it can, yesterday I went into the Korat area, the heavy infected area. We have the field nearby, heavy infected, the field of Rayong 72, the field of Kasetsart 50, still, field still look okay. If farmer, use not susceptible, but this is free, this 2 or 3 variety, and rope the infected plant at the early stage, 1 or 2 months. I think this method, and select the replanting later next season, select the clean planting material. I think in the heavy infected area, we can still produce cassava under heavy pressure of disease. So I for my recommendation, if you're scoring the disease this coming season, if have the percent infection, so I can use that data too. But I don't have the disease infection of the new clone from IITA, but I sure that, from your severity rating, that 5 clone from IIT will be very, very low, because very easy, I think very easy. Look at the healing capability that Xiaopeng show, the healing capability is very high. If we can select the resistance, I think not difficult, but how to combine, the resistance of high starch, high root yield. I think that is more difficult. Resistance source, I think not difficult, but it is hard. Only that information. Thank you.
Thank you, Dr. Chermsak. I think that's really important. We're sort of talking about the longer term solution, and in the short term, sharing a consensus on what varieties we should be promoting. As many people may know now, CMD's been found on the border with Cambodia in Lao. Fortunately, that area is mainly Rayong 72, but if it makes its way further north, the main variety is Rayong 11. So we should be working hard now to switch varieties before the disaster arrives, because it will. So these are important observations. Is anyone from I know Dr. He's online, or I'm not sure if Nhan or Tui have dialed in they're in the field at the moment scoring plants, but any observations from Tay Ninh about what varieties you see that are currently available for farmers that are that short-term solution?
Well, Dr. He, would you like to give some comments?
Yesterday I come to Thailand for Harvard to experiment. So most of the variety, all of the variety now have infection CMD, but some variety even have infection CMD, but still have a high yield around 50 tons per hectare. And the situation now is all of the variety is still low. So this year we have that the soil from I sent to us. We have 2 or 3 experiments. So most of the variety we import from CR now is low infection of CMD. But I think some is branching and some is germination is not very well. I think we have to take more and maybe we use the soil material and for roasting maybe for next time. I see. So now I think Asia and CR and other have about to have the good material or the future for resistant or tolerant of CMD. So we hope 1 year or 2 years more we have the good variety for our project. Thank you.
Okay, Xiaofei, I see your microphone is unmuted. Are you looking to make an intervention?
No, I think you know I'm pretty new, so it's really coming to still in the process to learn the situation. So it's I believe for now, based on our observation, probably KU50 and Rayon 72 is really showed moderate resistance for short term. It might be still good, the best option for farmers. But for long term, hopefully we can integrate the CMD resistance to this kind of high yield, high diameter clones. So yeah.
Dr. Augusto.
A question?
Yes, please, Hanan.
Hello to everyone. A couple of questions. I know that Chalermpol made a selection on the segregating families for CMD using the marker. Have you been able to validate if the marker properly selected materials carrying resistance? And the second question is if you have any timeline regarding the arrival of the materials from India.
So I believe Imra can give some information about the variety from India. I have several emails go back and forth with Imra. He is talking with the partners in India, so they can agree to how to say, to export, or CR can import the varieties. So we are working on the SMTA. So Imra, if you can, if I miss anything, can you please add some?
Thank you, Xiaofei. Thank you, Hanan. I have several discussions with Dr. Sheila. So, and we have come up with with 6 varieties that we want to, or lines that we want to bring in. And now we are at the stage of, we have also discussed that who is going to multiply and how is going to multiplying and how many plants we are going to bring in. So that discussion has been done. Now it's the preparation of the, we need to send the request, and the SMTA need to be finalized. I'm really hoping that by end of year it should be here in Laos or in Vietnam.
But on the Thai side, I know there's been high-level representation to the Indian government. Has that progressed? Has that been solved and the material is coming?
We are waiting for the answer from the ICA. ICA is Indian Agricultural Research Council. So we have the message from Dr. Sheila that we can get 3 varieties from India. But anyway, lastly, so we have to wait the MGA from ICA. So CTCRI cannot provide us the MGA, but ICA should do that. So we are waiting for the MGA from ICA. And I don't have the timeline because the people from the Ministry of Foreign Affairs from Thailand just talk to them, and we are waiting the answer. And but anyway, we already have the import permit to send to India, but we didn't get yet the MGA.
Thanks, Dr. Vichan. Jerome Paul, are you there and able to answer Hanan's first question? Maybe not online. Eric and Serena, any questions from Canberra?
Thank you, Jono. Well, first, I'd like to thank everyone for providing very precise and useful updates. And also, it's really a good achievement of this project to coordinate this sort of knowledge and the trials using the same material in many places for what clearly is more complex than we think. The difference between countries is a puzzling phenomenon, but probably explained by some biology of either the virus or the insect, maybe. So I don't really have questions. I find the good work going on, and as I say, lots of it. The question that for the future is, it seems like there are lots of irons in the fire, lots of opportunities to be explored, and so the challenge will be to prioritize them properly to reflect the resources we have and our partners have. And thank you for Vietnam to provide all that work to work with our project. It's really, really great to see. So the prioritization is how are we going to choose and follow on all the possibilities and more to come? That's the question I have, but of course that's all what the project is about.
Do you want to answer that one, Jono, first, or shall I ask a second question?
I don't want to answer it, but Xiaofei can answer it, and you can ask a second question.
Okay, so I think we have different source populations and we have different activities. So I think this is mainly determined by the the nature of breeding. So we mainly have 2 ways. One is try to introduce resistant variety. Hopefully we can find some good clones. The performance is comparable with the released variety under the CMD pressure. So if we are not that lucky, so so at the same time we are developing a breeding population by crossing CMD resistant clone with a lineage released variety. So we all these activity belong to these 2 scenario. Yeah, I think that's my my vision to group all these activities.
Well, I think it's worth mentioning that the crossing blocks in Vietnam have been established at elevation, and Hung Loc has established the red light system to induce for flower induction. So that work is ongoing to get those populations.
Yeah, thank you, John.
Jerome Paul, I can see you're unmuted. Hanan had a question about some of your earlier work? No, sorry, we're not hearing. Luis Augusto, did you have any comments as our fearless leader?
No, actually, I think the amount of new data coming in is overwhelming. I think that the work is going really well. I'm really pleased to see that we are actually collaborating with the national partners on putting genomic selection in the toolbox for developing new varieties. I think this is very encouraging for cassava, and to see that the different investments that we have done in the last 3 years or 4 years are paying off, including the work on selection of varieties in Vietnam, and also the work on the collection that is going to come to realization soon. So we are sending the germplasm collection for further screening, as Xiao Fei mentioned. So I think we are pretty well set up. So I was enjoying a lot the presentations from all the people, and I noticed the KU50 situation on the different testing blocks. So it would be nice to see the reason why we get that variability. So that would be my contribution. So thank you. No, thank you. Great presentations.
Kunvumi, would you like any reflection from the industry?
Thank you, Jordan. First of all, I'm very impressed to see the progress of other countries and collaboration with SEAT and other partners to do this progress. I appreciate very much. I think it is quite a critical to have this resistance variety, and we have to do all our effort to be able to survive the industry until the resistant variety available. But technically, I don't know much about the breeding. I can understand part of it. One of the point that Dr. Wu present, and one thing trigger in my mind is about the germination when he got the seed, and he got some variety have a good germination, some variety have a poor germination. In my experience, when I try to grow several variety, and I also see that when we use the cutting and the ability of each variety to germinate is different. So maybe I can just ask whether when you make a selection of the resistant variety, we look a little bit forward when we release into the industry or to the farmer, because the germination is not good. Then we might have the farmer might have they lost a lot of plants in the field. They have to replant, or some of them they don't do replanting. Then it will affect the yield. So that is the only technically that I can only add. But I think very important to say I appreciate all the breeders to have done this good progress, and I look forward to see the success soon. Thank you very much, and thank you, Jono, for organizing this.
Thanks, Bumi. Serena, I know you had a question waiting in the wings.
Yeah, I have a technical question if you can hear me.
Yes.
So I wanted to know a little bit more about the grafting. You've got a very clear system for scoring for disease in the field that's been used for many years across many different trials. So we kind of understand the pros and cons of that 1 to 5 scoring system, and you've got a level 2 which you've sort of set as being at some sort of theoretical threshold around whether something is sort of tolerant or resistant or not. Can that same scoring system be applied to that grafting in the field, or do we need to develop a different way of assessing that And my second or aligned question is, I don't understand exactly the process of the grafting, but I'm assuming you can't then take yield measurements, that the graft is different and that the yield would not be relevant data. Is that correct?
Dr. Vu, would you like to answer that? And maybe King?
Yes, so for the scoring on the grafting, on the grafted plants, we use the same system. So basically, if you see the leaf, how much it is deformed or the yellow color changes, then you can score them as if it's on the field. And yeah, so basically, if you see if you see this, yeah, so and on the yield data, so actually when we do the grafting, we graft on the on other plants. So actually, and we don't use all of the plants that we grow, so we can still do some scoring for the yield, because when we plant the clones of interest, we plant at the same time with the disease plants as the spreader. So we can graft them on this spreader plants. And if you're interested on the yield, then you have to plant more of the clones of interest. But if you don't want to score the yield, then we just plant enough, like say 5 or 10 plants. But if you want to score the yield, then you have to plant more. Yes.
Thank you very much.
Okay. Okay. So thank you very much, everyone, for joining. We're going to try and have a few more of these. I think one of the next ones will be really about the seed system, you know, looking at what varieties we should be promoting at the moment, how can we multiply them quickly to get them to farmers, where can we multiply them. We're also hoping to have one on to get a better sharing of information about the current status of not only where the disease is, but where it isn't, so we can identify those areas where potentially farmers could be sourcing planting material. Very concerned for the coming season in Thailand with all that area infected in Korat, where a farmer's going to get clean planting material in the coming season. So if I send you an email, I hope you accept to be a speaker in the coming weeks. Other than that, thank you everyone for joining. Thank you for those people who have stayed up in Colombia. And I look forward to seeing you all very soon. Thank you.
Thank you so much. Thank you. Thank you, Jono.
Thanks, Jono.
Thank you, Jono. Thank you for organizing. Very good meeting. Thank you.
Thank you, everyone.